Cell movements through body tissue are an essential aspect of many biological processes, including development, wound healing, and immune responses. There are several ways that cells can move through tissues, including 1. Amoeboid movement. This is common in immune cells, such as white blood cells. It involves the extension of the cell membrane in the direction of action and the formation of pseudopods, which are temporary protrusions that help the cell move forward. 2. Mesenchymal movement is seen in fibroblasts and some cancer cells. It involves the formation of long, thin protrusions called filopodia that pull the cell forward through the tissue. 3. Epithelial movement. This type of movement is seen in cells that comprise epithelial tissues, such as the gut lining or skin. It involves the coordinated action of cells, with the cells at the front of the group pushing forward and the cells at the back of the group pulling up. 4. Chemotaxis. This is the movement of cells in response to a chemical gradient. For example, immune cells can move toward areas of infection or inflammation in response to chemical signals released by damaged tissue. In addition to these mechanisms, cells can also move through tissues by squeezing between cells or through gaps in the extracellular matrix. The specific mechanism of cell movement depends on the type of cell and the tissue it is moving through. The cell nucleus does not directly produce enzymes that dissolve collagen. The production of enzymes, including those that dissolve collagen, is a complex process that involves multiple steps. First, the DNA in the cell nucleus contains the genes that code for the enzymes. When these genes are activated, transcription occurs, in which the DNA is used as a template to create messenger RNA, mRNA. The mRNA then leaves the nucleus and travels to the cytoplasm, used as a template to synthesize the enzyme protein through translation. Once the enzyme protein is synthesized, it may undergo further modifications and processing to become an active enzyme. In the case of collagen-dissolving enzymes, such as matrix metalloproteinases, MMPs, these enzymes are secreted by cells and can then break down collagen in the extracellular matrix. Therefore, while the cell nucleus does play a crucial role in the production of enzymes, the actual process of enzyme synthesis and secretion is a complex, multi-step process that involves many cellular structures and organelles. The production of enzymes in the cell is a complex process that involves several steps. 1. Transcription. The first step is the activation of the genes that code for the enzyme. The cell nucleus contains the DNA, which serves as the blueprint for the production of all proteins in the cell, including enzymes. When a particular enzyme is needed, the genes that code for that enzyme are activated, and the DNA is used as a template to create a messenger RNA, mRNA, molecule. 2. mRNA processing. The newly synthesized mRNA molecule undergoes several processing steps, including adding a 5-cap and a poly-A tail and splicing to remove non-coding regions, to produce a mature mRNA molecule. 3. Translation. The mature mRNA molecule then leaves the nucleus and enters the cytoplasm, where it binds to a ribosome. The ribosome reads the mRNA sequence and uses it as a template to synthesize the enzyme protein through translation. 4. Post-translational modifications. After translation, the enzyme protein may undergo several changes to become an active enzyme. These modifications can include the addition of sugar molecules, the removal of specific amino acids, or the folding of the protein into its active conformation. 5. Secretion. Once the enzyme is fully processed and activated, it can be secreted by the cell into the extracellular space or remain within the cell, depending on its function. In the case of enzymes that break down collagen, such as matrix metalloproteinases, MMPs, these enzymes are typically secreted by cells. They can then break down collagen in the extracellular matrix. Various signaling pathways regulate the production of MMPs, and their activity is tightly controlled to ensure proper tissue remodeling and repair. Secretion of enzymes into the surrounding tissue. Enzymes can be secreted by cells into the surrounding tissue through several mechanisms. One standard tool is secretory vesicles, membrane-bound organelles that transport molecules from the Golgi apparatus to the cell membrane. The contents are released into the extracellular space when the vesicles fuse with the cell membrane. Another mechanism is through a process called exocytosis, in which the enzyme-containing vesicles fuse with the cell membrane and release their contents into the extracellular space. This process can be regulated by various signaling pathways, such as calcium signaling, to ensure proper timing and location of enzyme secretion. In the case of enzymes that break down collagen, such as matrix metalloproteinases, MMPs, these enzymes are typically secreted by cells. They can then break down collagen in the extracellular matrix. MMPs are synthesized and processed within the cell and then transported to the cell surface or into extracellular vesicles for secretion. Enzyme secretion can be tightly regulated to ensure that the enzymes are released in the appropriate location and in the proper time. Dysregulation of enzyme secretion can lead to various diseases and conditions, such as cancer, where enzymes can contribute to tumor invasion and metastasis. Enzyme molecules making their way through collagen fibers of the tissue. Enzyme molecules can make their way through collagen fibers in tissue in a few ways, depending on the size and structure of the enzyme and the collagen fibers. One way is through diffusion. Enzyme molecules are relatively small compared to collagen fibers, so they can move through the gaps between the threads by simple diffusion. This can be facilitated by water or other fluids in the extracellular matrix. Another way is through proteolytic activity. Certain enzymes, such as matrix metalloproteinases, MMPs, can degrade collagen fibers through their proteolytic activity. MMPs can cleave the peptide bonds between amino acids in the collagen fibers, leading to the breakdown of the collagen structure and the release of individual collagen molecules. In addition, some enzymes can bind to specific sites on the collagen fibers, allowing them to interact with the thread and potentially modify or degrade it. For example, enzymes such as lysyl oxidase can cross-link collagen fibers by oxidizing specific amino acids. In contrast, other enzymes may cleave or modify specific amino acids in the collagen structure. Matthew, a cancer and stroke survivor, requests that you help spread awareness of severe diseases by publishing this video. Please take a moment to subscribe to and like this video.